from the University Herbarium, Karn Imwattana, who came to Duke from Thailand, and he's been studying peatmosses for last 5 years, and he's actually in his last year of graduate school, and he is using specimens from the herbarium for his research a lot, and he would like to show you what can be done with the records that you are transcribing right now and what he found on his quest to understand peatmosses.
Thank you, Blanca. And yes, today I will share with you the part of the research I've been doing, and the name of my talk is like tracing histories of plant using herbariums and field collections and genetic structures of some circumboreal peat moss. So, like organisms are not static; they evolve through time, and like as the time goes on from like deep history now to the present day, there can be several events that can occur in the histories of organisms. Like population can be split and diverge into different lineage or species. Some population might go extinct. They can be processed like bottleneck, where like population was like rapidly reduced and then expand again. And or like they can be like rapid population expansion. And all of these events in histories of organisms, they can be like correlated with the physical environments in the world. So like some events in the past, it can be like tectonic, like continents moving, or like climate changing, like physical environment, like variation through time. So like knowing like histories of organisms can tell us something about what is happening in the past and their response to it. So, and along this time, like they're like along, and what is great is that like along this evolutionary time, like the organism, they are continuously like writing their own history in their genomes, like along, like there will be like genetic mutation occurring along, like as the time goes on, and these are all recorded in the organism's genomes, and we can use this like genetic data to learn more about the histories of organisms. And yeah, and the organism I'm interested in is peat moss, which is a group of bryophyte that can be mostly found in wet area, like wetlands with low nutrients and cold temperature. So in northern hemisphere, like peat moss usually form this ecosystem called peatland, and actually they are one of the few bryophyte that can be like a dominant plant in the ecosystem. So in peatland, like most of the area is covered by peat moss, and they are ecosystem engineer. They can dominate the area, and they can they can grow a lot, and actually like they can store like a lot of biomass underground as well, because they are like constantly waterlogged, has very low oxygens, and the decomposition is very slow. So they can be like a very significant carbon sequestration area, which can be very important for global climate. And also like sphagnum is a very diverse groups of plant. Like around the world, there are like 300 to 500 species worldwide. And you can see from this picture, like they are like all kinds of color, all kinds of shapes and forms. They are very, very beautiful groups of plants. And yeah, and the groups of sphagnum that I am focusing on is called Sphagnum capillifolium complex, which is a groups of like 6 to 12 closely related species. And this is the picture of some of the species that I study. And what is interesting about this species is that there are several species in this group that have like circumboreal distribution, like they can be found all over northern hemisphere,
so I wonder what the stuff
on different continents in northern hemisphere. So like the distribution is very, very wide. So one of the questions that I have is that like for this species with like circumboreal distribution, is there any patterns of genetic structure within this species across the geographic range, like is like all the plants in the northern hemisphere, are they all similar to each other, or they are clustered in some way? And is there a common pattern in the genetic structure among these circumboreal species? And this will lead to the last question, which is, I think, is very important. Like, can these patterns of genetic structure can they be explained by any events that occurs in the past? Like, can we learn something about the histories of these peat moss, like from this genetic structure? And to do this, I need plant samples from around the distribution range. I need the sample from all the areas that they occur in nature. So in this study, I got some like field collections that I collect by myself in the field. But yeah, like since the distribution is so wide, like I cannot travel everywhere to collect the fresh samples. So as you can see, like I use a lot of herbarium collection, and this allow me to get like plant samples from around the world. So like this project will not be possible without like herbarium collection, because like you can see like all of the samples I have from Europe, from Siberia, or like from like very northern Arctic area, they are all from herbarium collection. So like this is herbarium, like what we are doing today is like very, very useful for doing research like this. So yeah, this is the sample that I use. And from the herbarium sample and like the field collections, I have to get the genetic data out of this sample. So this is just a short summaries of the process to extract DNA. I think it's really pretty to show it. So yeah, first you got this like plant samples in the test tube. You grind it into like fine powder, and you add some extracting solutions, and then you get the DNA. Like in the right picture, like you can see the white bands in the test tube. Like these are the DNA extraction, and these are the genetic materials of the plant, and then I will get like the DNA sequence out of this and do the analysis. Well, yeah, this is the lab work I have been doing. So today I will show you like one of the analysis I've been doing is called the analysis of genetic structure. So basically, I am trying to use this program called admixture to cluster the samples into genotype groups. So basically, like a sample from the same genotype group will tend to be more similar than sample from different groups, and some sample may be a mixture of different genotypes, which can so from this method, like we can group samples into cluster, and we can also know whether if there is like any samples that have like a mixture of multiple clusters, which can be a signature of like gene flow, like plants from different groups exchanging genetic material. So I will show you the result from one of the species called Sphagnum fuscum. Like this is a species that mostly found in like open peatland. They're forming like hummock, which is like a tiny hill, like tiny mound that is like raised a little bit above the water tables of the wetlands. And this is one of the main hummock forming species. And this is they are quite distinctive in the field in a way that the color is usually yellow or brown. So very easy to spot. And this is the genetic structures of Sphagnum fuscum. So you can see from this figure that we have two groups of samples. We have two groups of plants. The one group, the one represented by blue color, mostly centered around Eastern North America and Europe. So like plants in these areas are like they are very genetically similar to each other. And the other groups is centered around Alaska and Siberia and Japan. So this is species that these are plants that are around the Pacific Ocean. So and then there are the area between these two clusters, like the area around southeastern Alaska or somewhere around like in the middle of Siberia, that show some patterns of admixture that they have like genetic contribution from two groups. So yeah, this is the pattern we see in Sphagnum fuscum. One group around Atlantic Ocean, another group around the Pacific Ocean. And then, yeah, and when I look further into like the rest, like the 4 other species that I studied, they all show similar pattern. Like yeah, like you can see like the blue cluster, the blue group, like they all these species, they all center around the Atlantic Ocean, and the other group, the red group, they mostly are in Alaska or in East Asia or Siberia. And this is a very striking pattern that occurs in 5 different species, at least in my study. And this is common in other peat moss species as well. So yeah, so from this, well, you can see that plants in Europe and Eastern North America, they are very closely related, and they are both like very different from plants in Western North America. And how can we explain this pattern? Like one of the possible explanation is most likely explanation is that with this pattern, it suggests that these peat moss in Northern Hemisphere, they during the Ice Age, they might have been like divided into 2 populations in the ice-free area. One area is in Eastern North America and Europe, and the other populations in the Beringia. Beringia is the area like in Western Alaska and Eastern Siberia that is not covered by glaciers during the Ice Age. So this is the area that like a lot of Arctic plant has been resided during the Ice Age. And then after the Ice Age, when the glacier retreats, like the plants in these 2 refugia, they just expand their populations and colonize like the area that's been left free by the ice, and then they result into the distribution we see today. So this is one thing that we can learn from. And one more thing is that, yeah, and you can also see that the admixture, like the gene flow between population, they usually occur where at the edge of these 2 populations. So it is likely caused by gene flow by secondary contact. And this is one of the examples of the thing that you can learn from like using herbarium specimen, like getting genetic data, and we can compare them and we can learn about the history of plants. And yeah, and this is my presentation. Thank you so much. And if you have any question, feel free to ask. Yeah.
Super interesting. Thank you so much. I did have a question. So how easy is it to get DNA from a dried herbarium specimen? Is there like an age limit where it's too old and you can't extract it, or how does that work?
Yeah, I think like from usually like, yeah, old samples are very challenging, and like the oldest samples that I can get like good DNA from my like, because it's like around 20 years, and like and from that age, like I think like half of the samples that I try, like half of them didn't work. It's like so you can get some DNA, but it is more challenging than using new samples. And there are like several ways that you can like you might need to use more material to get like good amounts of DNA, something like that. But yeah, but it is possible to get like, yeah, for me like the oldest one I have is like 20 years or maybe like 25 years. And yeah, and I think this varies from like the groups of plant, and also like the technique you are using for sequence DNA too, because I'm using like the genome scale level sequencing, which like need a lot of some quite amounts of DNA. So yeah, you need so yeah.
Very interesting. Thank you so much.
Can I have a question, Karn?
Yes.
What was the most amazing place where you traveled to collect your peat mosses?
That's a difficult question. There are several. For me, I think like one of the most exciting places would be Barrow, Alaska. That is like on the North Slope, like next to the Arctic Ocean. And like one of the species that I study, Sphagnum warrenstoffii, they occur from like they can occur in Arctic area. And actually, like this is very fun species. Like they have very broad distribution. So like the So yeah, you can find it in North Slope, Alaska. And another place that I found it that I think really interesting is the mountains in North Carolina, which is like very southern. Like it's just amazing to think that I collect this species like from both North Carolina and Northern Alaska, and this is like very different latitudes. And yeah, it is. Yeah, that would be my pick for special place. Yeah. Yeah. Yeah, and for me, it's also really fun to look at the sample from Herbarium and see like where they come from too. Like just look at the map and the location that this specimen is from is also a really fun thing to do. And like, yeah.
A lot of nice comments and thanks in the chat, if you see that. Any other questions for Karn before we get back to transcription?
If no one else asks, Karn, are there peatmosses in Thailand?
Yes, actually there are some, mostly in the mountains, because like you need to go like high elevation to get like cold temperature. There are like around 10 species. They're not that much compared to like boreal area, but there are some, like mostly restricted to the mountain. I think this is the case for like most of the tropical sphagnum, like in Central and South America, they are all in the mountains as well. So yeah, there are some, but yeah, in high places. Yeah. Yes. Yes.
Righty, well, if there aren't any other questions, thanks again. A big thanks again to Karn for joining us today. And good luck with the rest of your of your research. I'm going to go ahead and stop the recording.